The first step was filtration, second is reabsorption, third is secretion, and finally the excretion or removal of the urine. Okay. In case of filtration, the main components required and the main things to be kept in mind was the renal blood flow, peculiarities of renal blood flow, okay, then the structure, okay, of the glomerular capillaries and the nephron, the starting part, together they form the Bowman's capsule, okay, and what the substances are taking the route, whether they are totally getting filtered, whether they are getting reabsorbed, or whether the substances is getting added to the filtrate. So depending on the three properties, finally the composition of urine takes place and it is excreted. Rate of urine formation is 1 ml per minute, okay, and parallel to the nephron, we have two types of vessels present. The two types of vessels are peritubular and vasa recta, depending on the presence or location of the nephron, whether they are present in the cortical area or in the juxtamedullary area. So this was just a brief introduction. In the last class, we have started with glomerular filtration rate. Okay. So what is the normal value of GFR? You get MCQ on this. Okay, if we are talking it in minutes, it is 125 ml, and if we are talking in a day, it is 180 liters per day, out of which 1 to 1.5 liter is getting excreted as urine. Okay, the important part to remember is you get MCQ again on this particular section, that what do you mean by filtration fraction? It is GFR upon renal plasma flow. Renal blood flow is 20 to 22% of the total cardiac output, which comes out to be 1200 or 300 ml. So out of that much portion, how much is the plasma present? So it is 650 ml or 700 ml. Okay. So GFR, we know the value 125 ml and we know the value of renal plasma flow. And with this formula, we get to know that it is 0.2 or 20% of the plasma is getting filtered. This particular fraction is known as filtration fraction. Okay. You must be at least clear with the terms. जब अपने पास क्वेश्चन आता है ना, तो हमें उस चीज का आना चाहिए. ये इसलिए इम्पोर्टेंट है. यू गेट अ क्वेश्चन, डिफाइन ग्लोमेरुलर फिल्ट्रेशन रेट, गिव इट्स नॉर्मल वैल्यू, add a note on estimation of glomerular filtration rate, plus describe in detail factors affecting glomerular filtration rate. So एक बहुत लेंथी क्वेश्चन हो गया और इसको अपन ब्रेक करेंगे, तो यू गेट 4 short notes. Clear? So you have to prepare the topic according to the need of the question. Okay, and you get 4 or 5 MCQ on this particular section. And whenever we complete renal system, okay, I'm going to give you a Google questionnaire. Okay, you have to fill that. We'll get a feedback that how much you have learned or how much you have tried to gain information or knowledge. Okay, whenever the topic gets over. Okay, this is the structure of the filtrating membrane, the 3-layered structures which was present. Depending on the structure of the membrane, the important things were the slit pores or fenestrations that was responsible for filtrating the structures or the substances, plus the charges. Okay, the collagen and the proteoglycans which are present in the basement membrane, they are mainly formed of negatively charged substances, so they repel the negative ions. So the proteins do not get filtered through this particular filtrating membrane in normal healthy conditions. Okay, if they are present in the filtrate, it is proteinuria, but it is normally not present. So you get question, explain why they are not getting filtered, because they are getting repelled by the negative charge present on the collagen substances and proteoglycans which are present on the basement membrane. Important question regarding this is, you might get a short note on estimation of GFR. Okay, it is the gold standard test when we are talking of the renal function test, which gives us the idea of glomerular filtration rate. Okay, to go for this test or estimation, a substance has to have certain criteria which need to be followed by the substance which we are using for this estimation. इस GFR टेस्ट को करने के लिए हमें जो सब्सटेंस चाहिए उसको 4 या 5 क्राइटेरिया फॉलो करने हैं. उन 4 या 5 क्राइटेरिया में से सबसे इम्पोर्टेंट है, इट हैज टू बी फ्रीली फिल्टर्ड. व्हाट डू यू मीन बाय फ्रीली फिल्टर्ड? the substance which is present in the plasma. When we are talking of filtration, now we are specifically talking about the renal plasma flow. Okay, we have segregated, we have separated. We are not talking of renal blood flow, we are talking about the renal plasma flow. When we are talking of renal plasma flow, the filtrate which is coming, okay, so sorry, these are the conditions. It has to be freely filtrable at the glomerulus. So the size of the substance, the charge of the substance, okay, the solubility of the substance, it has to match the criteria, and the substance has to get filtered easily through the filtrating membrane. It has to enter into the Bowman space. Clear? Another feature required is it should not get reabsorbed. Reabsorbed means when the filtrate is coming in the tubular section, okay, parallel to the tubular portion, we are having another set of vessels that may be peritubular, that may be vasa recta. So the substance has to pass from the tubule into the interstitial space, from the interstitial space into the blood or the vessels which are present. So they need not to go again to the blood, so उनका reabsorption नहीं होना चाहिए. Secretions means they should not be added to the filtrate. So 3 criteria you have to keep in mind that is required for GFR estimation. Not synthesized by the tubules, that is they should not be added by the tubules, not broken down by the tubules, or metabolism should not take place. So these criteria are met by a substance that is known as inulin, which is we get from a plant dahlia, okay, and this substance is infused, okay, it is not present in our body endogenously, but it is exogenously given, okay, infused in the body. So this gives us the exact estimation of GFR. But when we are talking of clinically, the another substance which is used for GFR estimation is creatinine. Okay, so the formula for estimation of GFR is, okay, clearance. That is this particular concentration of a substance, how much time it takes to clear, okay, from the body. So this is done with the help of 2 substances, exogenesis, inulin, endogenesis, creatinine. From where creatinine is coming? From the breakdown of the creatine present in the muscle. So it is present endogenously, we need not to infuse. But inulin, we need to infuse. Okay, the best result we are getting from inulin. Okay, but clinically, creatinine is considered as the gold standard test for GFR estimation. Okay, but when we are going for creatinine GFR estimation, some part of the creatine is being added through the PCT segment of the nephron. तो जो GFR की वैल्यू इन्यूलिन से आ रही है उससे 5 to 10% increased value आएगी when we are going for creatinine GFR estimation. और ये fluctuation क्यों हो रहा है? क्योंकि some part of the creatine is being added from which part of the nephron? From the PCT segment of the nephron. So there is variation in GFR 5 to 10%, and that variation is due to this secretion of the creatinine from the PCT segment of the nephron. Okay, so this about clearance, you get a short note, or if you are quite lucky, you might get a lengthy question on this GFR, define, give its value, okay, clearance and factors affecting it. Okay, so you have to read your topic according to the question also and according to the requirement of MCQ parallel. Okay, so whatever the substance is getting filtered, so at the time of filtration, the rate at which the glomerular capillary is filtrating all the blood at a particular time, that is glomerular filtration rate. Okay, Px or Pin, okay, when we are talking of inulin, it is in, or any other substance creatinine X, Y, Z, so we can put a value X. So Px is the concentration or the amount which is present at the time in the plasma. GFR is the rate at which it is getting filtered, glomerular filtration. So whatever the amount is getting filtered has to be excreted. So this is the formula, concentration of inulin in the plasma into GFR equivalent to the concentration in urine into the rate of urine formation, V. So with the help of this formula, we calculate the GFR. This is in case of inulin. When we are talking of creatinine, okay, it is the normal breakdown product of creatinine and endogenous compound that fulfill these criteria, but there is variation due to the secretory part from the PCT. Okay, so there are fluctuations in this case. Okay. Now this is about the clearance. Next topic we have to cover is about Starling forces or the factors affecting glomerular filtration rate. Okay. So last class I have talked about 4 types of pressure. Hydrostatic pressure and colloidal osmotic pressure. Clear? Now, how they are going to affect the glomerular filtration rate and renal blood flow. If you remember, we have talked about the renal blood flow and its peculiarities, that it is an auto-regulatory mechanism and what all things are required for this auto-regulatory mechanism. Okay, when we are using normal value, it is this net filtration pressure. You have to remember 10 millimeters of mercury, and it is a filtration coefficient having a constant value of 12.5. So this filtration coefficient depends on 2 things most important. First is thickness of the filtrating membrane. जो filtrating membrane था ना, 3 layered structure, उसके ऊपर filtration coefficient depend करता है. तो ये MCQ आ सकता है. You can get an analytical reasoning if there is change in the thickness of the filtrating membrane. The thing which is getting affected is the filtration coefficient, and if this filtration coefficient gets affected, it is affecting the glomerular filtration rate. Secondly, it is getting affected by the surface area available for filtration. Okay, so 2 things which are affecting the filtration coefficient. One is the thickness of the filtrating membrane. Second is the surface area available for filtration. So if there is increase or decrease in the surface area, that is definitely going to affect the glomerular filtration rate. The thing another which is getting affected, the GFR, is the net effective pressure. और net effective pressure को हमने कैसे calculate किया था? क्योंकि इसके एक-एक forces के ऊपर पूरा question आएगा. The factors affecting GFR. So the formula is GFR is filtration coefficient and net effective pressure. Net effective pressure we are calculating with the help of Starling forces. The 4 forces which are required for this net effective filtration pressure is the capillary hydrostatic pressure, Bowman's hydrostatic pressure, colloidal osmotic pressure of the glomerular capillaries, and colloidal osmotic pressure of the Bowman's space. What forces favor the filtration is the capillary hydrostatic pressure, which is highest. Okay. What forces inhibit the filtration? The hydrostatic pressure present in the Bowman's space. Clear? Another forces which inhibit this filtration is colloidal osmotic pressure of the glomerular capillary. And last but not the least is the colloidal osmotic pressure in the Bowman's space, which is 0 normally, because the filtrate lacks the plasma protein. So there is no colloidal osmotic pressure seen in the Bowman's space naturally, normally, physiologically when we are talking of. So whenever there will be fluctuation in either of these pressures, there will be change in the net effective pressure, and when there is change in this, definitely it is going to affect the GFR. But the renal circulatory system has a command over it, that whenever there is change in the mean arterial blood pressure. We are not talking of the blood pressure, but we are talking of the mean arterial blood pressure. When you're talking of blood pressure, it is simply systolic and diastolic, but when you're talking of mean arterial blood pressure, it is diastolic blood pressure plus 1/3 of the pulse pressure. What is pulse pressure? It is the difference between the systolic and the diastolic blood pressure. So with the help of two pressures, we can calculate other two pressures, that is the pulse pressure and the mean arterial blood pressure. So whenever there is change or fluctuation, okay, our blood pressure का effect देखेंगे when we are walking, we are talking, we are at rest, we are having change in posture or exercise, किसी भी चीज का अपन change होता है तो blood pressure change होता है, but it has a reflex mechanism that it becomes normal within seconds. So if you do not take the recordings or observation within 10 seconds or 20 seconds, the fluctuations will be gone. Okay, so this autoregulatory mechanism works especially in the renal system. So whenever there is fluctuation of mean arterial blood pressure from 80 millimeters of mercury to 180 millimeters of mercury, the blood, that is the renal blood flow, remains same. Why? Because it has to maintain glomerular filtration rate. If there will be change in the arterial pressure, there will be change in the net effective pressure, there will be change in the glomerular filtration rate. So to regulate the glomerular filtration rate, the renal plasma flow or blood flow is regulated. It is regulated auto-regulatory mechanism. Two supportive mechanism are given. We'll see to it. और ये कब तक कंट्रोल में रहेगा? When the mean arterial pressure ranges between 80 to 180 millimeters of mercury. Clear? अब अगर उसके extreme change हो रहा है blood pressure में, then it is definitely going to affect the glomerular filtration rate, because there are physiological changes. Okay, blood pressure अभी पढ़ लिया आपने theory CVS में? No, you are still on ECG, I think. So when you are going to read about the blood pressure, the factors affecting blood pressure, okay? So 24 hours after meal, okay? After sleep, during sleep, after change in the posture, running, walking. So there is fluctuation in blood pressure, but they are physiological changes. They are not pathological changes. So within a particular range, the glomerular filtration rate is maintained until unless it meets at the extreme end. So the range for the autoregulatory mechanism is from 80 to 180 millimeters of mercury. Within this change of blood flow or the arterial pressure, the GFR remains constant, that is 125 ml per minute or 180 liters per day. So the factors affecting is broadly filtration coefficient and net effective pressure, but net effective pressure is calculated with the help of Starling forces. And what are the main four Starling forces? The hydrostatic pressure and the colloidal osmotic pressure. So whenever there will be fluctuation in these pressures, definitely there will be change in the GFR. Okay. This is filtration coefficient, hydraulic conductivity and surface area. Normally not highly variable. Diseases that can reduce the filtration coefficient and GFR is chronic hypertension, when there is increase in blood pressure, that is chronic for a longer duration. Obesity, diabetes mellitus or glomerulonephritis. Itis means inflammatory conditions. Bowman's capsule hydrostatic pressure कब change होगा? Normally changes as a function of GFR, not a physiological regulator of glomerular filtration rate. ये कब change हो सकता है? जब tubular obstruction हो, filtration possible नहीं हो रहा है. Okay, kidney stones, tubular necrosis, urinary tract obstruction or hypertrophy of prostate or cancer. So these are the conditions in which there may be change in the value of Bowman's capsule hydrostatic pressure. That is, the filtrate is coming into the Bowman space, but it is not getting allowed to enter into the loop of Henle or finally into the further distal parts. Okay, so these obstruction will change the बोमेंस हाइड्रोस्टेटिक प्रेशर, एंड बोमेंस हाइड्रोस्टेटिक प्रेशर क्या करता है? इनहिबिट कर रहा है, फेवर कर रहा है? इनहिबिट कर रहा है। तो ये इनहिबिट करेगा तो जीएफआर की वैल्यू कम हो जाएगी, है ना? तो अब अगर आपके पास में क्वेश्चन आता है एंड यू आर लैकिंग विथ टाइम, so you can give your answer in a flow chart fashion, but your arrow should be proper, your nomenclature has to be proper. Whatever you are writing in short form, please make it a complete form, okay, and make it in a clear handwriting so that it becomes clear. Arterial plasma autocontact pressure, okay, if it is increasing, it is increasing the glomerular pressure, that is the filtration, arterial plasma oncotic pressure. क्या होता है ये? Colloidal plasmatic pressure of the afferent arteriole or the glomerular capillaries. ये बढ़ेगा तो फिल्ट्रेशन बढ़ेगा या कम होगा? कम होगा। तो इसके लिए आपकी हर किताब में विच एवर बुक यू आर कंसर्न, अरे जिस भी बुक आप कंसल्ट कर रहे हैं, हर बुक के अंदर चार्ट दे रखा है, बट देयर माइट बी टाइपिंग एरर्स ऑल्सो, so it is always better to look for 2 books and add a note, sticky note in your primary book whichever you are focusing on. Clear? एक बुक को बेस बनाओ, दूसरी बुक से क्रॉस रेफरेंस करके उसके स्टिकी नोट बना के आप अपने उस पर्टिकुलर टॉपिक पे स्टिक करो, so when you are going for final revision it is easy for you to revise through a single book, एंड में कभी भी जाके उस बुक को चेंज मत करना। Filtration fraction was GFR upon renal plasma flow. Okay, if there is increase in the filtration fraction, it is going to increase the GF. Okay, approximately this is net filtration pressure. This is hydrostatic pressure of the glomerular capillary 60. Okay, this is 28 pressure. Glomerular 60 है और यहाँ पे क्या हो गया? 36 हो गया। क्यों हो गया ऐसा? यहाँ पे तो आपका कोलोइडल ऑस्मोटिक प्रेशर ग्लोमेरुलर कैपिलरीज का 28 है और यहाँ पे 36 है। ज्यादा हेल्प मत करना, ठीक है? अपन अभी 1-2 महीने में करेंगे हियरिंग टेस्ट, so I can hear your whispers, okay? At one end, when we are talking at the end of afferent arteriole, the colloidal osmotic pressure of the glomerular capillary is 28. जब अपन efferent arteriole की तरफ आ रहे हैं तब यहाँ पे 36 लिखा है। So what might be the reason? बड़ा होता है, ठीक है? और you cannot say that it is bada hota hai, you have to say that the diameter of the afferent arteriole is bigger. Okay, if you are saying bada hota hai, then you get 0.5. Okay, what is the next probability? Because the post lunch session is a graveyard session, so we have to be interactive. तुमसे वो पूछूंगी जो तुम्हें नहीं आता होगा। पार्ट्स, पार्ट ना, पार्ट। दो 3 महीने लगते हैं, मुझे याद तो हो जाते हैं नाम बच्चों के, है ना? नाम रोल नंबर याद हो जाते हैं। क्या हो सकता है? थोड़ा सा रीजनिंग लगाओ अपनी। कोई बहुत ऐसा डिफिकल्ट क्वेश्चन नहीं है अपना ये केबीसी का या किसी का? The only difference is the concentration and the amount of plasma protein which is at the afferent end, at the efferent end. When all the plasma has been filtered, water has been filtered, solutes have been filtered, so what remains and reaches at the efferent arteriole is the concentrated plasma protein, which is responsible for tremendous increase in the glomerular colloidal osmotic pressure at the efferent end. Clear? So it is not a single slide or a single graph. You have to explain it or give in detail that what and why are these values are seen on the slide. Okay? And this is the hydrostatic pressure of the Bowman's space. Plasma protein concentration, arterial plasma oncotic pressure, if it increases in the efferent arteriole, it is going to increase over here. So, now glomerular hydrostatic pressure. What are the factors which will govern this or control this? What are the factors that can change the hydrostatic pressure of the glomerular capillaries? कौन कौन सी चीजें हो सकती हैं जो कि इसके प्रेशर के अंदर वेरिएशन ला सकती हैं? The first and foremost important thing which we always remember when we are showing the diagrammatic representation of the filtration or the glomerular structure, juxtaglomerular apparatus, okay, or ultrafiltration, the one thing which we keep in mind is the diameter of the efferent and the efferent arteriole. So whenever there will be change in the diameter of the efferent and efferent arteriole, it is definitely going to affect the change in the hydrostatic pressure, but this tremendous change is regulated or maintained with the help of auto-regulatory mechanism, though until unless the extreme change in the pressure is seen. Extreme change कहाँ है? 80 और 180 के बीच. अगर 80 और 180 के बीच में फ्लक्चुएशन कुछ आ रहा है मीन आर्टीरियल प्रेशर का, तो जीएफआर में या हाइड्रोस्टेटिक प्रेशर में ऐसा कोई चेंज नहीं आएगा और जीएफआर की वैल्यू कांस्टेंट रहेगी. बट अगर 80 से कम 70, 60 हो गया तो इफेक्ट में आएगा. ठीक है, और ये कब होगा? जब चेंज इन द डायमीटर ऑफ द एफरेंट एंड efferent arteriole. Not only the change in the diameter of the efferent and the afferent arteriole, there are certain hormones, there are certain chemicals at the neuronal level which are responsible for the change in the diameter. So indirectly it is controlled hormonally plus neuronally. So it is not only the constriction and dilation of the vessels, afferent and efferent, but this constriction and dilation is brought about by two important factors, hormonal or neuronal. And until unless there is extreme change in the pressure, there will not be change in the hydrostatic pressure of the glomerular capillary. ठीक है? So factors that influences arterial pressure, effect is buffered by auto-regulation. Clear? So auto-regulatory mechanism work करेगा. किस रेंज में वर्क कर रहा है? 80 to 180. अब आप कौन सी बुक को डील कर रहे हैं ये आप देख लेना. But maximum, please try to refer Guyton for these values. You are not able to challenge your answer until unless it is given from Guyton or Gano. Clear? क्या आपकी जैन के अंदर ये इफेक्टिव फिल्ट्रेशन प्रेशर में हाइड्रोस्टैटिक प्रेशर 45 मिलीमीटर ऑफ मर्क्यूरी भी दिया जा सकता है. Guyton में it is 60 millimeters of mercury. So whichever book you are dealing, but please for units and values go for Guyton or Gano. Okay. Afferent arterial resistance and efferent arterial resistance. This is arterial pressure. If it is normal, then 60 to 180. Okay. The GFR, the pressure remains the same and it is the नॉर्मल किडनी, ग्लोमेरुलर हाइड्रोस्टेटिक प्रेशर की वैल्यू कितनी रहती है 60, और आर्टीरियल प्रेशर अगर नॉर्मल रेंज में है, ऑटो रेगुलेटरी मैकेनिज्म की रेंज में है, तो 60 ही रहेगा। और अगर अनहेल्दी किडनी है तो किडनी डिजीज के केस में देयर इज चेंज इन द ग्लोमेरुलर हाइड्रोस्टेटिक प्रेशर। क्लियर? Okay, this is mean arterial pressure, auto regulation of renal blood flow and GFR, but not urine flow. और प्लीज रिमेम्बर, याद रखना इस बात को, आज भी कह रही हूँ मैं, और नेक्स्ट आपके सेमेस्टर में इसके ऊपर जरूर क्वेश्चन आएगा, बट व्हाट आंसर वी गेट इज व्हेनेवर वी गिव द क्वेश्चन ऑफ फिल्ट्रेशन, 90% of the students, okay, they write we are filtrating urine. We are not filtrating urine, we are filtrating blood. अभी बोल रहे हो पर ये हर साल की कहानी है, ठीक है ना, हर साल की कहानी है। एंड देन दे कम एंड आर्ग्यू दैट मैम मार्क्स नहीं दिए बिकॉज यू हैव रिटन ब्लंडर। वी आर नॉट फिल्ट्रेटिंग ब्लड, वी आर फिल्ट्रेटिंग प्लाज्मा, रीनल प्लाज्मा फ्लो। बताया था ना अभी, अभी बताया था ना, हाँ या नहीं? तो अगर आप उसको रीनल ब्लड फ्लो लिख रहे हो देन यू गेट 0.5, but when we are talking of filtration, what we are talking of? Renal plasma flow, है ना? उसके बाद में भी मैं तुम्हें 110% गारंटी दे सकती हूँ कि सेकेंड सेमेस्टर में you get this question and 90% of the students are going to answer this. This is, ये राइटिंग में दे सकती हूँ मैं। So effect of afferent and efferent arterial constriction on glomerular pressure, if there is increase in the resistance, okay, there will be decrease in the renal blood flow, which is going to decrease the pressure, hydrostatic pressure, that is going to decrease the glomerular filtration rate. क्या टाइम हो गया? 2:41 में ही तुम बोर हो गए? यस, तो फिर चलो खड़े हो जाओ, तुम बताओ। इधर से बहुत आवाजें आ रही है, इधर से पूछते हैं ना, इनको बहुत आता है। खड़े हो। योर नेम? सारांश? हाँ सारांश, क्या होगा अगर अपन इफरेंट आर्टीरियल का रेजिस्टेंस बढ़ा दें? इफरेंट आर्टीरियल की भी अब बात करें, इफ देयर इज इंक्रीज इन द रेजिस्टेंस ऑफर्ड बाय इफरेंट आर्टीरियल, क्यों? रीनल प्लाज्मा फ्लो कहाँ बढ़ जाएगा? रीनल प्लाज्मा फ्लो रूप ऑफ हेनले में कहाँ आ रहा है बच्चे? किस चीज की कंसंट्रेशन? जब अपन रीनल filtrate की, filtration is a process and what we are getting for the filtration is filtrate. So the filtrate is entering at what point? हाँ, कहाँ एंटर होता है फिल्ट्रेट सबसे पहले? पीसीटी। वुमेन्स कैप्सूल किस चीज का बना हुआ है? व्हाट आर द कंपोनेंट्स ऑफ वुमेन्स कैप्सूल? So that is why I am asking, the filtrate gets entered where? filtrate कैपिलरी में आएगा? कैपिलरी से तो फिल्टर हो रहा है, तो वो कहाँ एंटर होगा? What do you mean by Bowman space? Nephron is the blind funnel shaped structure is the beginning part of the nephron. that blind funnel shaped structure is forming the Bowman's capsule, and the inner part of that particular is known as Bowman's space, where the blood is getting filtered or the renal plasma is getting filtered from the glomerular capillaries. It will enter into the Bowman's space, the funnel shaped structure. That Bowman's space is continuation with PCT. ठीक है. अब ग्लोमेरुलर कैपिलरीज का नेक्स्ट सेट ऑफ कैपिलरी नेटवर्क क्या होता है? It is a dual set of capillary network. So if the nephron is present at cortical area, what is the another set of capillaries which are present adjacent to it? Peritubular capillaries. So before the peritubular capillary, what is the structure present? That is the efferent arteriole. So what happens if there is increase in the resistance? That is, the efferent arteriole is constricted. So what will happen to the GFR? डिक्रीज होगा. क्यों? Decrease होगा, but when the resistance offered is of severe type. If it is a moderate type resistance or given to the vessel, efferent arteriole, what will happen initially? Whatever the blood or the plasma which is coming for filtration, that has been accumulated in the glomerular tuft. So initially excess amount of plasma is coming, so excess amount of filtration is taking place. So initially the GFR will increase, but if the duration of this constriction increases and the severity increases, then there will be decrease in the GFR. तो इनिशियली इंक्रीज होगा, बट ड्यूरेशन अगर लंबी रही, सीवियरिटी लंबी रही, तो जीएफआर की वैल्यू कम होगी. अब क्या होगा, आप लोग एग्जाम में लिख देंगे दिस इंक्रीजेस जीएफआर, दिस डिक्रीजेस जीएफआर वैल्यू, बट देयर इज नो एक्सप्लेनेशन गिवन फॉर दिस. ओके, एंड स्टिल यू एक्सपेक्ट marks. So if you have completed your assignment, you know what was needed in the paper and what you have written in the paper. Okay, so don't get bored in the class. We just try to give you a gist. ये बहुत लंबे लंबे topics हैं. We are just trying to give you a gist. You have to go and read. Until unless you are going to read, your concepts will not be clear. Okay. So if there is increase in the diameter of the vessel, okay, there will be increase in the hydrostatic pressure, more of the blood is coming, so more of the glomerular filtration rate there is, because increase in the resistance offered at the efferent arteriole. Okay, तो ये इनिशियली होगा. तो जब आप बुक पढ़ोगे तब आपको पता चलेगा कि ये इंक्रीज क्यों दिखा रहा है, and after some time it will show the value decrease. Yeah. Summary of the determinants of glomerular filtration rate. Filtration coefficient increases, okay, then GFR increases. This is hydrostatic pressure of the Bowman's capsule. Okay, so there will be decrease in the GFR. Colloidal osmotic in the glomerular capillaries, decrease in the GFR, but उसका explanation होना चाहिए. ठीक है, आपके बुक्स में ऐसा टेबल या पिक्चर दे रखी होगी, but you have to explain. This is efferent arteriole. Okay, filtration fraction. Okay, as long as increase in the efferent arteriole resistance 3 to 4 times of the normal. So they have written the condition. Okay, इसका मायोजेनिक मैकेनिज्म नेक्स्ट क्लास में करेंगे अपन. ठीक है. हाँ या ना? उसके साथ में हम पढ़ के आएंगे ट्रांसपोर्ट मैकेनिज्म, एक्टिव, पैसिव, difference between primary active and secondary active transport mechanism, transcellular pathway and paracellular pathway. Clear?